Most welcome my friend in our channel. We are going to start some scientific research update on the experimental model Lac Operons. This is the type of the genetic regulation pathway where this experimental pathway shows that how prokaryotic animals, prokaryotic organisms regulate their gene expression. What is the means of Lac? What is the means of operons? You can understand operon means a group of genes. There is a number of the groups that can operate under the some influence of the molecules. Lac means here is the lactose. Lactose is a molecules that is disaccharide sugar molecules that can act as inducer molecules in the some animals like eukaryotic, not the prokaryotic animals like E.coli, Yhr coli. Lac Operon for studying gene regulations. Lac Operon is the Lac pathway where switch off, where switch ons. We need to understand both pathway. When Lac Operon, this model is going to switch off, switch on. Generally there is present in the mediums the E. coli. This pathway is switch off because there is no lactose, so the pathway is switch off. Now we understand how many number of the genes are regulated because operon is the group of the genes. There's two type of genes are presented in operons: controlling genes and structural genes. Controlling genes, regulator genes is called represented by the I, promoter genes represented by P, and operator genes represented by O. Now we are coming to understand what is the means of structural genes, that mean that can express the protein, that is called structural genes. These are also three types: Z genes, Y genes, and A genes. Now we are coming to understand regulator genes. What is the means of this regulator gene? That is also called inhibitor genes. Note they can represented by I. Not here you can understand I is not the inducer. This is the I represented inhibitor, not inducer. Transcription and translation, we understand when they produce the repressor proteins, when there is a presence of not presence of lactose, this regulator genes can reproduce a proteins is called repressor proteins via the transcription pathway and translational pathway. When Lac Operon switch off, the active proteins bind with the operator genes. I will show you in the diagrams. When there is no presence of the lactose, this Lac Operon is switch off and this repressor proteins bind with the operators, so there is no transfer of the RNA polymerase from the promoter to structural genes. Regulation of the Lac Operons by repressor is also called as the negative controls. Why it is called negative, we will discuss later. So in these reasons, operator genes can not bind or structural genes not regulated by the operator genes, and this is proteins is not structural genes. That means when there is no presence of the lactose, they cannot bind with the repressor proteins, so the repressor proteins are free, they can bind with the operator, and there is no structural proteins are forms because though there is no any signal. Promoter genes, what is the means of P is also promote, they can produce after translation transcription pathway. RNA polymerase joins the structural genes and where they start the formation of the structural proteins. Operator genes, I already told you, operator genes and repressor proteins are both bind and on and off when there is lactose presence or not. Structural genes, I already told you how many number of structural genes: Z genes, Y genes, and A genes. Z genes forms the after transcription translation pathway, they produce beta galactosidase enzymes, and Y genes produce the permease enzymes after translation transcriptions, and A genes produce the transstylase pathway. Now we will understand diagrammatically so you can understand better way. When this is the type of the fragment of the DNA where large number of the other part are presented, they are going to transcription and translation pathway. When the lactose is not present, the Lac Operon is switch off, absent of lactose. I already told you lactose already disaccharide. When the beta galactosidase enzyme presence, like glucose and galactose, they form the monosaccharide. These are these are the molecules that can be used by the bacteria for their growth and other sexual process. So lactose is used for the energy purposes. If there is a present, then they will act to this pathway. This is called controlling genes, where the I already told you. This is called inhibitor proteins, promoter and operator. All these are controlling genes are presented. Structural genes ZYA. So if there is no lactose is present, then it will go for just like this is a membranes of bacteria, plasma membranes. This is a medium. Here there is no lactose. So this repressor proteins, this called is the inhibitor proteins. They start producing a transcriptional process, repressor mRNA, and repressor mRNA going to produce after translation process repressor protein. These proteins, active proteins, get bind with this molecule, this pathway, this region of the DNA. This is operator region. Operator, if this is bind, then there is no promoter can produce RNA polymerase that go for these regions. So repressor bind with the operator and prevent the transcription pathway. There is no transcription pathway because this protein is going to bind here. So the promoter, which is going to produce RNA polymerase, this polymerase is not transferred, not going to give a signal this region. So there is no transcription, no translation pathway. When Lac Operon switch on, that means in this medium, there is a medium, this is the plasma membranes, and presence of the glucose, lactose is here. Lactose is going to allolactose. I wrote here glucose, this is not glucose, this is lactose and allolactose. When this protein molecule, this region and inhibitor regions produce the transcription process, mRNA, repressor mRNA molecules, they go for the repressor proteins formation after translation. The lactose permease enzymes present, generally small number of enzymes are present permeases. They transfer from the outside to inside by this process. Permease help, and they bind with this repressor proteins and form the allolactose molecules. So this repressor active proteins are not free, go to bind with this region. So the promoter is free, and they will start producing RNA polymerase, and RNA polymerase transfer their signal to structural proteins, and these structural proteins produce like mRNA molecules, and these due to transcription process, and they are responsible for production of the beta galactosidase enzymes, permease enzymes, and transstallase enzymes. One by one, Z produce beta galactoside, Y produce the permease, and this produce the transstallase enzymes, and they are going to break down for the lactose into the monosaccharide. I already told glucose and galactoside, and bacteria are free to use these molecule monosaccharide for their growth and other purposes of sexual lives. So this is the whole process of the molecules that can produce switch on, switch off lactose pathway. So you can understand when the Lac Operon is on, when Lac Operon is off. If there is a lactose is present, then is off, not off, on. If there is no, there is a off. So overall is this molecule lactose is showing Lac Operon pathway expression of the prokaryotic genes, how they regulate their expression of these molecules. Hope you like this video. If you really like, please